Hello everybody, my name is Alejandro Gonzalez and today I will present this work called First Record of Bilateral Hypertrophy in Uca rapax in Laguna Madre, Tamaulipas, Mexico. As an introduction, let's talk about the distribution of Uca rapax. First of all, Uca rapax is a field crab with a wider distribution worldwide at tropical and subtropical areas. The distribution comes from Florida, USA to Santa Catarina, Brazil. Specifically in Mexico, his distribution comes from Laguna Madre in Tamaulipas all over the way to Quintana Roo, expanding the distribution described by Thurman in 2018, because we also found it in Tamaulipas and Veracruz. However, this specific Uca rapax was located in Laguna Madre, Tamaulipas. Ossipodids from the genus Uca present a marked sexual dimorphism, which is easily visible as the male develops a major claw which can reach up to one-third of its total weight, while the other claw remains reduced. This is known as heterochelity. The major claw represents a secondary sexual characteristic in males, which play a major role in the specific sexual selection and the male-male intersexual interaction. The minor claw is also important for feeding and burrowing. So the heterochelity is very important in these organisms. However, several cases of these abnormalities are registered more often each time. As we can see in the following table, which is a modification of Valeria Fonseca et al. At 2015, in which they registered 11 cases of abnormal chelipeds. Then Da Silva and Shinosaki Mendes at 2018, who found Uca rapax 2. And finally, in the present study, we also found a hypertrophied Uca rapax. These are some images from other authors. Uca rapax was collected in Isla de Barrera, a southern area of Laguna Madre, Tamaulipas, as a part of a study of diversity of Uca species in the region. This organism was placed in a bottle with 96% alcohol for preservation. At the Laboratory of Ecology at the University Autónoma de Nuevo León, the individual was identified and measured with a stereoscopic microscope and added to the carcinologic collection at the same university. Later, we applied the man-Whitney range test for two independent samples at the data collected from the measures. Finally, we took an approximately 100 grams of mud to analyze the percentage of sand, silt, and clay. As results, Uca rapax with bilateral hypertrophy collected in the previously mentioned area is considered the first registered in Mexico with this condition. We didn't find any significant difference between the measures we made, so this confirmed that the chelipeds are subequal, like we can see in these pictures. The unusual morphologies had been recorded by several authors. However, the main question here is why is more frequently the cases of this abnormal and what are causing it? Ahmed at 1976 realized that this condition can be an abnormal case affecting an embryonic or chromosomal level, and authors like Morgan in 1920 that could be genetic alterations. Furthermore, other authors like Ferrier in 2015 mentioned that could be a dysfunction of androgenic gland that affects the hormone expression, causing an overexpression. Considering these aforementioned possible causes of this condition, we now need to think about what is triggering the overexpression or the mutations of fiddle crabs. Could be the changes of the environmental conditions like pH variation, contaminations, heavy metals, hydrocarbons, and other man-made impacts in the ecosystem causing this? So we need to continue looking for answers using new methodologies like genetics, ecophysiologic, endocrinological analysis, and environmental DNA analysis, so we can understand the causes of these more frequent conditions. Finally, we would like to thank to the members of the Ecology Department of the School of Biology at the Universidad Autónoma de Nuevo León. and also to the members of the carcinology department at the same university. Thank you very much.